people with exam, with this.
Yes.
And people, so, yes, so.
She said it,
Because we have in the semester.
Can I? But if it's recorded, then people can watch it. We don't have topics.
This is a workshop. How many time you need to? I'm not sure. 2 hours?
We can have a break.
So the idea is when you are.
A jacket if I get cold. Yeah, so I've broken it up into 3 sections, mostly because they're very different stages. And so I want to do, if people have questions, because I've been getting a lot of questions already. So then we can go through if people have questions at each little section before we add more content, especially with the analysis, because if I want to address any questions in like primer design before we get into the deeper stuff. Okay, where did I put that pointer? Okay, so, all right, so we're going to talk about, this is getting sort of into the details of eDNA, how what is it? And how do we actually process these samples and analyze samples in the lab? I'll be giving a seminar tomorrow for the faculty, and students I assume are also invited, where I talk more about the applications of this technology. But I know you guys are doing genetic work, and so I'm going to get into the weeds here. So we're talking about this emerging field of eDNA science, and why should you learn about this? This is going to become more and more and more prevalent in the fields of molecular biology, molecular ecology, bioinformatics. This is up and coming. And so it is a good idea, in my opinion, for everyone, ecologists, molecular biologists, and conservationists, to have some knowledge, at least what is this? Because so what the goal is here is for you to understand a little bit about what eDNA data are, because I think we're going to move into an eDNA genomics era, probably in the next decade. As soon as technology advances, this science is going to advance as well. So I've got 3 different parts here. First, I'm just going to sort of set the scene. And I know I've already been getting a lot of questions from some of you about how do you get DNA from water. So this is going to be a little shorter. And then we're going to talk about how we can analyze and how we design the assays. And then we'll actually talk about the data analysis in a little bit more detail. We'll have a break here. If we need to have a break here, we can also do that. But I want to do questions in between these sections. So this is, informal. If you have a question or something isn't clear, please ask. Okay, so environmental DNA. What is it? All organisms are shedding DNA into their environments. So for a fish, it could be scales, it could be mucus, waste, blood. It's all going into the environment. Right now, you are also shedding DNA, skin cells, hair, okay? And so it becomes environmental DNA once that DNA is in the environment. And we can collect this eDNA, this environmental DNA, when we collect water samples. eDNA is not limited to aquatic samples. So one way to think about this is it's like a forensic science. So if you left, we could take samples and maybe see who was in this room. So it's very forensics, very CSI techniques, but we're just moving it into the water. There's also, what else is emerging is they're getting environmental DNA from snow and they're melting it. Air, so they're taking air samples and actually capturing DNA aerosols. So one way to think about this is instead of getting your DNA derived directly from tissue or an organism, is you're pulling it from the environment. And that's why the applications of this are extremely far-reaching. So there's in terms of the analysis, there's two very broad scale approaches. The first one is metabarcoding. This is where you take, I'm going to stick with aquatic here, you take a water sample and you screen it for everything that is present in the water using some sort of universal primer. So I'm assuming this is probably similar to the work that you do perhaps with your microbiome stuff, where you are screening for maybe all the fish, for example. The other main approach is called targeted species detections. This is where you're interested in particular species. It might be one species, it might be two, but here you have to design a species-specific assay that's going to detect the DNA that you were interested in, and then it ignores everything else. The bulk of eDNA research uses metabarcoding. In my lab, we use targeted species detections. So that sort of sets us apart from a lot of the eDNA labs that are out there. You might say, why? Don't you get less data with that? It depends on the question that you're asking, because this is great if you want to know about diversity, biodiversity, if you want to know all the different kinds of things. But as you probably know, there's issues with universal primers. If you have something that's taxonomically distinct, it won't detect it. Or if you have rare target among a lot of common non-target, for example, or a rare species amongst common species, you might miss it because of preferential amplification of the more common species DNA. So for the types of questions, so you know, I talked to yesterday about what my lab does. We work on rare species on the verge of extinction. So that is why we use targeted species detection. It's much better at finding rare species. So this is the general workflow. And then we're going to break down these parts. We conduct eDNA field surveys. So we collect and filter water. We extract DNA from the filters. And then we screen for target DNA using PCR. The benefits of this, why is this emerging? Why are we seeing this become more and more common? It's more sensitive than traditional methods. So yes, you can put a net in the water, but you're probably going to miss a lot of things. eDNA is better at detecting species. This makes it cost effective when you have a species that's in low abundance. If you have a very common species, yes, if you put a net in the water, you're likely to catch it. But when you have a rare species, the cost of all of those people and all of that time and the likelihood of finding it, eDNA is more cost effective in this arena right here. And funders love cost effective. It's also non-invasive. You know, if you want to go and handle an animal or capture it in a net, that can be a very stressful experience for the animal. And we know now that handling can cause stress that can, you know, reduce immune function and cause other health issues. And we don't need permits. This is probably one of my favorites because I ship things internationally all the time, and it can take years to ship tissue. For this, we're just shipping a filter, which you'll see, and that doesn't require any permits. So what are we using it for? Again, you have to remember, this is just a tool. It's a forensic tool, and this is not the limit here either. So you can look for invasive species, protected species. You can use it to monitor, look at seasonal migration patterns, spatial patterns, changes in distribution with climate change, species abundance, historic trends in abundance. Early days, but there are some population structure studies out there. You just have to think about it as in like you have the potential to sample an entire population from water samples. In my lab, we do research on all of these topics, and I'll be talking about some of them tomorrow. Today, we're going to talk about the actual science, though, a little bit more. So what are the challenges? So eDNA is tough. It's tough work. The first study was published in 2008, freshwater species from small ponds. It's not that hard. If it's freshwater in a small pond, it's a contained environment. And then it started to become more popular, but it really didn't take off in the marine environment for maybe in the last 8 to 10 years only. One of the biggest challenges, especially in the marine environment, if you think about it, that they're shedding DNA, but it's not huge amounts of DNA that are being shed. So what happens is we've got a low number of copies of DNA, and there's, depending on where you are, sunlight, UV, degrades DNA. It's just in water. It's not preserved. It's not like a nice tissue sample that you get that's preserved. And there's microbial decomposition occurring, which also degrades DNA. So it's low copy, and it's getting smaller, smaller, and smaller, and smaller, and smaller as time goes on. So the chances of getting your target decrease as the time increases from shedding. But that's not all. So here's the degradation, but it's in the water, and especially in the marine environment, which doesn't have boundaries, it's moving around. So you start to think about how on earth can this possibly work, that you stick your bottle in the water and you actually capture what you're looking for. The other challenge, contamination. This is perhaps the biggest issue in eDNA work. What do I mean when I say contamination from eDNA? I mean contamination from target species. So we did some work on bull sharks many years ago now, bull shark eDNA. My number one rule is the target species are not on the boat. And somebody thought I was crazy and just over the top. And so we did an experiment, and they sent me a bunch of samples, and they said, I brought a shark on the boat, you tell me where. And I analyzed the samples, and I said, there was no shark here. After that, this is where the shark was brought on, and these are all positive. And it was exactly correct. So my rule is no sharks on the boat, because a shark on the boat is flailing around, it's spraying water everywhere, and you have DNA aerosols. You can also, though, get contamination between sites. Maybe this matters, maybe it doesn't. It depends on the question you're asking. If you were looking for something that is so rare it's never been recorded in the wild, maybe this doesn't matter so much. But for most of the work that we do, it matters very much. So what could happen is if you go to one site, and let's say the species is there, and let's say it's the marine environment, this is not contained, wave splashes. The risk is that if a wave splashes on the boat here with target DNA, and then you go to your second site here, your site 2 sample could be contaminated from DNA at site 1, even in the absence of whatever it is you're looking for. So this is a big issue. Notice I don't even put in human error, because then there's human error in there too. Okay, so how do we deal with this in order to have good science? Because it's very important that the science is still good. We use extremely rigorous protocols. We have, we've been, how long has it been? 8 years. We have been refining our protocols and our methods, and knock on wood, in the beginning, we had lots of problems with contamination. And so we'd go in the field, we would collect samples, analyze them, and we would say, we have contamination in the field. And we'd modify and modify and modify. Finally, we have something that works, that is simple to train others to do, and we get very little contamination now. If you deviate from the protocols, it's all bets are off. You might get contamination. Yes.
I don't understand. What is exactly the concept of negative control in EDA?
Yes.
What's exactly?
Mm-hmm.
I mean, what is one negative control?
That's an excellent question. So in our field work, we've got what we call negative field controls. What we do is we take the cleanest water available.
Yeah.
Where we are in the world depends. Ideally, autoclaved nanopore water. And we bring it into the field with us in one of these Nalgene bottles here, and we treat it exactly like a field sample. So it stays in the cooler with the field samples. If depending on our collection method, I'm going to show some different ways that we collect. But one of them is like a sampler, and we will pour the negative control into the clean sampler and run it through. So, and then we'll close it and put it back in the cooler. So we treat that sample exactly like we do field samples. And then we filter it, and then we extract the filter, and then we PCR it. That's just in the field. If we collect samples and then bring them back to the lab and filter, we will have a collection negative and a filter negative. And then we have a DNA extraction negative, a PCR negative. So at every single stage, we have a negative control. That way, when we have contamination, we can pinpoint, ideally. It happened, actually the most common place it happens is DNA extraction.
Okay, I understand this, but I think that there are a lot of bias in the design of the get sample, because
Yep.
Imagine
You're correct.
I can get sample from some side in the seawater.
Mm-hmm.
I am in the boat, you know.
Mm-hmm.
So there are many conditions that can change the Monday to Tuesday, you know. So
Correct.
the sample from the Monday is just a picture
Mm-hmm.
of this moment.
Correct.
So
So we
understand
take,
what is the diversity can change from the 1 month from after month.
correct. So that comes down to sampling regime. And your sampling regime, which I'm going to talk about, depends on your question. If your question is biodiversity, you need to perhaps go out there, you know, how many samples are, you collect much more than one sample. This is not one sample. You look at volume, I will get to this, but it depends on, you got to figure out the volume. How many samples? What seasons are you sampling? How many years of sampling are you going to do? So
and
it's
the deep in the water column.
Yep, we do that. Mm-hmm. Yeah.
Okay.
Yeah. Yeah. No, these are great. These are exactly the things that when we first started in this field, very little was figured out. And everybody was just sort of like flailing around trying things, and nobody knew what worked. And so we kind of went very, my approach to things tends to be extremely methodical, sometimes over the top. And so we broke down every single step, and we tested variations of virtually every single step to come up with something that works by and large. There's a lot of issues. There's a lot of it. It is extremely complicated. Absolutely. So this field work, that's why it took us 8 years to figure it out. Sterilizing things once isn't enough. So you may know if you autoclave something, yes, it kills a lot of the DNA, but it does not kill 100%. So we use multiple sterilization approaches in order to decrease the likelihood of contamination. Everything is cleaned and turned over at each site. So every time you're handling a new sample, you have new gloves. We use a lot of bleach. If we have waves coming on the boat or spray coming on the boat, depending on conditions, we will bleach the entire boat. We have mops, and I get the weirdest looks when I go on the boat because most of ecologists on the boat are filthy and they're dirty and they're just making a mess. And we're kind of the opposite. Again, no target species are allowed on the boat. And we bring water onto the boat with us, and we take this negative sample, treat it exactly like our field samples. I see no point. A lot of people will just bring a negative control on the boat, put it in the cooler, and it never comes out. That doesn't tell you. I want to know. So I'm not, sometimes people with their negative controls try not to contaminate it. And that's not the point. The point is, are the protocols we're using sufficient to prevent contamination? I want to know, is there DNA on my boat? Is there DNA in my sampler between samples? For the lab analysis, we also, and this is not complete, this is just to give you an idea. So we have eDNA lab spaces that are physically separated from gDNA workspaces. We have separate spaces for DNA extraction. We have separate spaces for PCR. We have unidirectional workflow. Our field equipment are in a completely different building even. So, and what this means is that I use a lot of space, which some people don't like. We're building a new building. And so I have 7 lab spaces in that new building. They're each small, but they're, and it's going to be very nice. I'm very excited, but this is what we need to have. If you don't have this, your data are not robust. Again, everything is sterilized at least twice. For our assay design, we will often use this tagged synthetic G-block oligios. So these are oligios just like you order your primers, except we mark it as synthetic. And the way we do that is we take a section of the fragment and we just invert it. And that way, if we start getting something weird, we can sequence it and know it's from this G-block. We use aerosol tips for everything. And so this one might sound strange. We don't have a positive control in our PCR as per ancient DNA protocols. So this one is specific to our lab because we use ddPCR. And I'm going to explain what ddPCR is. It's kind of like the next step above qPCR. The reason why we don't have a positive control is because with ddPCR, you cannot sequence the product. So if we have a positive control, it could contaminate the samples and we can't sequence it to see. So you do use a positive control if you're using qPCR or conventional PCR. And then of course we have negative controls. So these are just, I've got signs up like this. You're not allowed to eat fish. Well, you're not allowed to have food in any of my labs, but my students eat, we have like hall space that they eat in, and they're not allowed to have fish out there. No one is allowed in my labs just because I don't know what's on their clothes, right? We have a lot of ecologists, so we just have very tight regulations, and we bleach everything. So this is us bleaching kayaks that we thought we might need for sampling. And this is bleaching a bigger vessel. We just have sprayers for the bleach. The other way we negate the issues and the problems is we have optimized and tested and tested and tested for over years methods to yield the best results. So everything is fully optimized for low copy, low quantity target DNA. All right, so let's talk briefly about how we collect these samples. I've been getting a lot of questions about this. So this is what I usually put in a presentation and move on. But sampling regime. This is much, as Rob was just saying, this is much harder. On the surface, it seems super easy because people will say to me, hey, you just go down to the bay and I'll grab you 2 samples. And I'm like, no. I really wish there was some, and I feel like this is possible. It's just not my space at all. But I feel like there must be some sort of computational way to figure this out, to say what is best likelihood. And we are doing some work in detection probabilities, which, that will helpfully inform. How many samples? There's no magic in for population structure work. It's usually 30 per population, 30, 40 per population. It usually depends on how much money you have, frankly. How much money do you have? And then you max out that budget. What is your sampling regime? Are you going to collect randomly? Or are you going to collect in transects? For our marine work, we do most often, we do randomly generated sites within some sort of specification, depending on what we're looking for. So we might define an area. If the species we're looking for, we know uses waters of less than 10 meters, we'll make a grid over the area that we're sampling. And then we make microgrids, and then we have a random number generator that tells us which microgrid to sample in and which exact GPS coordinate to sample. But the biggest question is, what are you going to do? Maybe you can collect 50 samples. Are you going to collect those at high density in a small area? Or are you going to cover a larger area but not have such highly dense samples? Again, this often comes down to the question. However, we usually aim for good coverage over smaller areas, because my opinion is that if you're over larger area and you get all zeros, but you don't have a good density of samples, those zeros become less meaningful. If you sample one area really well, those zeros start to mean something. Replicates. How many samples are you going to take at a site? 1? It's not great. But if you want to take 3 samples at a site, you're probably going to be limited in how many sites you can go to. Depth. Surface, bottom, something in between. This usually depends on, everything comes back to the question, but if you're targeting a benthic species, one that lives on the bottom, you should be doing bottom water samples. We also know that eDNA is binding to sediment and other particles in the water, and it has vertical movement, so it is sinking. And we did some tests early on to look at eDNA at the surface versus at depth, and there tends to be higher densities at depth. But again, we were looking at a benthic species. So, and what depth? Bottom, mid column, 100 meters, 50 meters, depends on the species. And the volume and the pore size, 1 liter, 3 liters. I have seen, I do not do less than 1 liter. There's a lot of people out there doing 15 milliliters. I am not kidding. In a little hand syringe, I just think that is atrocious. So, oh, this is going online, I forgot. But more volume is better, right? More volume is better. But if you have a larger volume, you've got to think about your pore size. So we will typically use anything between, and this just depends on the habitat and the clarity of water, but we usually use between 0.45 microns up to 5 microns. And if you want to do 3 liters in 0.45 microns in a very turbid water, you can absolutely do that, but you have to have patience. That could take 8 hours, and you have to have the money to use a lot of filters. So the pods I'm going to show you, those are $20 US each. So if you have to use 5 of those per sample, that's $100 just for that US dollars. So it can get expensive. So you have to find this balance. It's you know, the volume and the ideal thing is to use the smallest pore size possible. That's what you want to do. So how do we collect it? We've done it every which way. When we first started, we didn't know very much, and we tried surface samples, and those results were okay. They're definitely easy to collect. But when we started doing some sawfish work and some other work on species that use the bottom, we needed a sampler that could collect water at the bottom and that we knew wouldn't accidentally collect water on the way down. So we needed to be able to deploy something that we could then open at depth and pull up and not have any leakage. So one of my students actually designed this and built this sampler. I'm going to show you a Niskin in a second. We did not use Niskin because these are not deep water habitats. This might be 2 or 3, 4 meters deep only. You're not going to use a Niskin at that depth typically. So I have a video just that shows how this works. Play thing. So here he's deploying it. He's just, and this is heavy. This thing weighs 40 pounds. I don't know how many kilos it is, 20 something kilos. So when he pulls these, he's opening the compartments, which are 1.5 liters each. The bubbles are telling you that you're collecting the sample. And we tested this thing like crazy before we used it. Here it's just draining off extra water that it took. And then it's got these spigots, and then we collect our sample. This sampler would be entirely turned over and cleaned between sites. And so for our negative control, we would pour it in here and run it through to make sure there wasn't any DNA residually in here. And we even tested our cleaning method to make sure it was solid. We also, if we're doing mid-water collection, we use these big Niskin samplers. These are deployed off of larger ships. This is a postdoc researcher in my lab, and this was actually for the Seriola project that we have. These are 10 liters each. So here she was collecting 10 liters in triplicate for each site. So we've collected the water, and then what are we doing? We've got filters to filter. We will typically use nylon or nitrocellulose. Yes, the material matters very, very much. Nylon performs the best in terms of DNA yield, but it really slows down filtering like crazy. And nitrocellulose is used more frequently in the literature because of this. And then we have these pods. So I have some here. So these are absolutely amazing. We haven't always used these. These just came on the market 1.5 years ago, almost 2 years ago. I love these. Yep. Yep. Oh, I'm going to pass it all around. He's going to filter. One of them is going to filter some water for me. So what's really cool about this, it's got a rubber top. And then once we filter a sample, I'm going to pass it around so you can actually open it and look at it. look at the pod. It has the filter membrane, which is whatever material and size you want. And then it has just a backer that helps with flow, a steel mesh part. But this is the prize right here. This bottom dries the filter. So you back in the day, this was so hard to do and not contaminate, because we used to, if we were filtering in the field with other types of pods that didn't have this desiccating bottom, we would have to open the pod in the field, and it's potentially windy, rainy, right? And we'd have to roll it up in the field and put it in ethanol. And that was very cumbersome. And that's why I don't mind spending $20 on each of these. I think it's a bargain. And what happens is after you've done your sample, you put it back in the bag, you remove the air, you ziplock it shut, and it is stable at room temperature for 6 months. And that desiccating bottom pulls out any moisture and preserves the filter. The DNA we get from these pods is so much better than what we ever got with ethanol. So here.
Yeah. It's very interesting this. This is possible to concentrate here DNA from water, but DNA of virus or bacteria in the filter here, and check the filter and do it the DNA extraction from the filter.
Yep.
Great.
Yep. And I have thousands of samples. Yeah. Smith Root is the company.
Of course, it's not Smith.
You're right. You're right. And I have asked them before if they will ship like to Indonesia, and they wouldn't. Okay, so this is in Panama. This is the setup of the filter, but I've got it here to show you how this actually works, and I want you to see the filter. So this was work we did in Panama last year. Our funder was amazing, and he put us in this really beautiful place in Western Panama, this huge mansion, hired a chef for us, and we had this pool area to do our filtering. This is from conservation funding. Like, it's just, so when you meet the right, it's all about who you know, at least with funding in the US. And this, I love this group. I love this guy. He's very passionate about shark conservation specifically, and I've gotten him to be pretty keen on shark genetic things. So that's been really great. So yes, this is me working hard, but I am really sweaty. So you can see, you know, I needed the pool for my breaks. But I've got 2 going here, and you can see I have separate pens for each sample, separate gloves for each sample. These samples took a long time to filter, so I would get one started, and then I would start another one, and it would be maybe anywhere for these samples, because it was mangroves, anywhere from, for 3 liters, 30 minutes to 3 hours to filter. And my budget was basically to get to only use 2 of those packs for each sample. So I'm going to show you how to use this, and how to do this. So who wants to do it? Do I have a volunteer? You guys can both do it. These 2 already volunteered that if we do some eDNA projects in Chile. Okay. So obviously in the field. Yes.
Another question before
yes.
Start the guys there. That's your talk is very interesting for me. What did your experience with the evaluator of the project about eDNA? Our experience in Chile with a proposal about this issue.
Yep.
Is very bad, because we receive a lot of negative comments of the evaluator about not sample size, sample distribution, but very negative. So I don't know if what is your experience
Yep.
with your peer review in USA. This proposal is the same, is very negative? Or
It depends on the qualifications and I guess the amount of detail they put. So I frequently review eDNA grants in the US for the federal and state governments. And it's one of those things where once you have a track record in eDNA science or someone on your team has experience, it becomes a lot easier to get the grants. So there is a lot of, I know in the US, there's a lot of bad eDNA science. Let's just start with that. There's a lot of stuff that I would not use the data for. And the government needs to make sure, at least in the US, that the data they're getting, they can use for whatever purpose they want. And the purposes are very wide ranging. So I would say my first, actually, USM, I funded my first eDNA project through funds from USM. Got that published in eDNA, the journal eDNA, which is pretty good. And then I published a couple more. And after that, I had sort of set up a reputation in this field in the US as doing very good work. So if I'm writing an eDNA proposal, I make sure I hit points about what am I going to do to ensure the data are robust, reliable, what controls are in place, is my lab set up for this, and the expertise. So like the more detail-oriented stuff. For sample sizes and things like that, I always say it's better to get more samples over a smaller area and make the budget work for that. Yeah, I think having someone on the team who has publication in this field is very important, because I rarely see projects get funded with no previous experience and really vague methods, or methods that don't make sense. So methods that are like, we're going to use this, and I'm like, or other reviewers are like, why would you do that? That's not, there's better methods out there, kind of thing.
Yeah, my conclusion is that here in Chile, there are no expertise necessary to evaluate this proposal. Of course, you have the expertise, but here in Chile, there are a lot of opportunities to apply this option of the study. For instance, will be interesting, maybe would you try to evaluate in the southern of Chile, where is the farm of salmon farm. There are a lot of problems with the farm, with the microalgae.
Okay.
Not even microalgae. So there are huge mortality of salmon every summer.
Oh wow.
Every summer.
Okay.
With a microalgae. But the problem is there are good macroalgae and there are bad macroalgae. But the problem is who knows what macroalgae is bad. Taxonomy study. This area is very important. eDNA. So it's a good proposal for you. There are a lot of money there, but the people from the farm, the salmon, have a big problem.
Okay.
I don't know some project or paper or idea about applying eDNA in the southern Chile.
Yeah, I don't know that much of this work has been done here at all in this country, which is why it's good for folks to learn more about it, and also identify good versus bad projects or methods when you read papers. Okay.
Go ahead.
All right, we're going to do this. This is your training for when I come back. Okay, so obviously normally we would have sterile procedures and gloves and we'd be bleaching everything. But for this, I just want to show you, because I know you were asking me, how long does it take? It must take, I think I asked you, how long do you think it takes for a liter? And you said 7 hours. So I have two. These are from, and there's a reason why I got, you'll see. These are from Quintero. I got these on the weekend with some assistance of some kids at the beach. I put them to work. So it's very clear. So anytime so for example, if I was to say, I want to do an eDNA project in Quintero Bay.
That is the water?
Yep. Yep. The first thing I need to know is what volume, what pore size. Because again, remember, I want the smallest pore size possible. So the first thing I always do when I get to a new site is I do a test. So just by looking at this, I brought 0.45 and I brought 5 micron pore sizes. I know this is way too clear for 5. So we're gonna, I have 2 samples and we'll do it with 0.45, and I think that's gonna be good. If it goes super fast, then 0.22 might even be okay. All right, so you're gonna set this up. So this is our water pump. It's a vacuum pump, it's very strong. I charged it last night. It's made for remote work. It's made for working on boats. It's very small. They can sometimes be tricky to fly with. So they're fine to fly with technically, but sometimes I have to open up the bottom of it and show them the inside. So because they're like, what is this? It does have on it flow rate. It's not the most accurate. So we would always measure our volumes. These are 1 liters. But it gives you some indication, and then it also tells you the battery charge. So the water, they're going to put this pod together, and oh, and you can adjust the flow rate here as well. You're going to, yep.
I have another question.
That's fine. That's why it's a workshop.
What is your recommendation? Make the filter instead in the field, or wait to come back to the laboratory?
That's a great question.
What is your recommendation?
So if the water is really, really clear,
yeah,
my recommendation is filter on site because it's very, very quick. If the water is very, very turbid, it could take 6 or 7 hours. And so in that case, that's usually when we're at some alternative site, which isn't our lab at USM. We will set up a filtering space wherever we are. So this was my filtering space in Panama. Nothing else happened here except filtering. It was bleached between every sample before and after as well. We also I have a separate lab at USM if we're going to filter at USM. But we honestly rarely filter there because our sites are usually several hours away.
Okay. Another question in the same idea is, imagine if you take a sample in one remote place and your lab is 3 days, 3 days of the far away, you know. So you need to add some preservative to the bottle or no?
So you have. Yeah, that's a great question. You have 2 options. So you do have to keep them in a cooler on ice until you filter them, because that DNA is degrading. And as soon as you bury them in ice and the temperature starts to drop, the degradation rate slows, right? So if you were 3 days away from the lab, I would actually, depending on logistics, recommend filtering on site with these. This company is amazing. They're actually I buy these from Australia. Yeah. They also have, I haven't tested it yet, but they say it's in development and they're happy for me to test it. They have solar powered chargers. So you could, my mic went out. Oh, there we go. So you could charge it using solar power as an option. About a 20-minute charge gives you 45 minutes of filtering. So, and then if we usually have multiple pumps in the field, so we'll usually bring three pumps, maybe 4. Two will fit in one of these cases. The other option, let's say for whatever reason, filtering is not an option. You would have to freeze the samples. Now, whether or not that is viable, is probably less viable than filtering on site with one of these. We used to have a very clunky filtering setup before these only came out 2 years ago, and it wasn't possible to filter remotely at that time. So this equipment has allowed us to go further and do more for sure. Okay. All right. So you can open that. I have another one. Where's my second one? Okay, pull out the contents. Yep, since we're not using it, so there'd be a whole lot of other rules about where things go and what you touch, but this is just a demonstration. Okay, so we call this a snorkel. You can put that on the white end. Everything is designed so that it only fits one way. Okay, and then this goes on the yellow end. Okay, and so what's going to happen is you're going to put the snorkel in the water sample, but before you do that, okay, we're subsampling at each stage in the analysis, which you'll see. And so we want to try and make that water sample as uniform as possible. So you're going to mix it. And that's going to get, if any sediment has settled on the bottom, which it hasn't, but you would resuspend it and try to keep it relatively even over the filter. So you can mix it, and then you can open it. Okay, and then you can put the siphon or the snorkel in there. Okay, now we're not going to turn it on quite yet because I need to warn you, this is very strong. So it might need to be, sometimes you have to prime it, kind of like an aquarium. So we're going to see if it needs to be primed or if it's very happy right now. But you're going to need to hold these bottles, okay? And I want you to look at the flow rate. And this one, you just want to make sure there's some air going into it. So you can hold it, just don't hold it tight, okay? So then you can turn it on when you're ready, which is right here. And then you can adjust the flow rate. Okay, so we're going. Can you move your hand? We're going. And you can see this one is filling up. Yep, so you can play with it. See, it's got your volume here. This one is actually, this is the fancier model. It's much quieter on your ears. The other ones, the base model, are very loud. They don't have this flow rate. Yeah, you can crank it all the way up and see what it can do if you want. Yeah. Okay, so let's turn it off. We're going to do a second liter because I'm hoping it'll clog. Okay, so I'm going to mix this, so this is our outflow. So we're actually just we wouldn't do this in the field. We're just going to move this to the outflow. Let's see if we can get 2 liters off of this pot. And you can leave it cranked up. We do it at max when we're in the field. Okay. So it's going quite fast, you can see. So usually we play with the flow depending on how turbid the water is. So it's really, really turbid and brown. Okay, so it's, yep. All right, so what we normally do, now we're gonna lift this up. What we normally do you have steady hands from all your PCRs? The last bit we can't quite get because it doesn't suction. So you're going to you're going to do it, but I'm just going to show. You're going to pour the last of it in like that. Okay. You have to clean all of this. So what you change the hose, and then we also put it in 10%. We get a bottle of 10% bleach, and we run bleach through the entire system. And then after bleach, we do water. Yep. And then, so you could wipe. We also have like stock and bleach wipes. I love bleach wipes. And we wipe the entirety of the entire workspace is bleached between, and the machine is also cleaned in between. You have to, so I will be cleaning this afterwards because it's got seawater and it could rust as well. Okay, so there's still one more step, and this is the hard step. Hopefully you have strong thumbs. Okay, you have to dry this filter. I'll do it first so I can show you. So you put your thumbs, and you can see, no, there's no water. Sometimes there's water there, but you put your thumbs here, and you need to crack it. You don't want to, it's okay if it happens, but you don't want to open it so hard that the filter goes flying, because then you have to throw it in the garbage. But you just crack it, and it can be kind of hard. And you know you've cracked it, you see that little lip, and you can hear it. So you do this for 30 seconds, and you're drying the filter then. But I'm just gonna pour some of this water in here because this is bubbling over. Okay. Make sure you're pointing this up. Use those thumb muscles. It's kind of an odd sensation because it's like you have to really push, but it's very pinpointed pressure. There you go. So then you hold it for 30 seconds. You can give him a try since he was laughing at you after. Make sure after you shut it and then let him do it. Okay, you can close it and then have him do it. Because you were laughing. Oh, that's so fun. There you go. It does get easier after it's been done a couple times. Now this water was really clear. You can do it for do it 30 seconds. This filter, I don't expect to have a lot on it because the water was clear. I'm going to show you photos of filters from turbid water. Very different. I showed you yesterday. Okay, that's probably good. Okay, so then what you would do is you would take this apart. These you recycle these, or you can reuse them if you bleach them and wash them. This would go back in here, seal it up, and then it's good for 6 months. So that makes this very user-friendly in remote areas. You can do this in places without electricity. If you have a car battery, you can charge this. And that's the point of, we're trying to develop and streamline methods to be able to work in places without electricity and things like that.
So the idea is you make this linear expression for the filter?
Yes. So there's like nothing on this, unfortunately, because the water was good. There is something on it, you just can't see it. So this is the filter right here.
Yeah.
And then this is just a backer to protect. Now, what else? The other thing, okay, one thing I don't like is you can't recycle this. This is compostable. So I compost this at home.
Perfect.
So I have thousands of these in my compost. And they're working because I said, this is really bothering me, the plastic, and say they're working to make this component compostable as well.
You say how many times you can leave this filter in the bag?
6 months.
6 months.
And then you can move the freezer, or you can move the filter to the freezer.
And don't happen nothing with the DNA?
Nope. The desiccation of it works brilliantly. It's great. I'll show you. I have extraction photos of the gels.
It's very similar. Time ago, we used the same bag with the blood from human.
I
don't know what is the exact name of the filter. This one is called. You can send this code back. There's no problem.
Okay. Yeah, you can ship these. Yeah. So I will ship cases of them back and no permits. Tissue samples, you need a permit. But there's this to them, if you just tell them it's water filtrate, and they're like, that's fine. That might change in the future. We don't know. But at the moment, there's no
Do you have
permit.
Some experience with the EDNA back from bacteria?
No.
With the methodology?
Not yet. I haven't done that. Lots of folks are interested in it, and I always offer my samples. So once the DNA is extracted, it's in my freezer. So I've been going to conferences and presenting on the data I have, the samples I have, and going, if anybody wants some DNA, go for it. So it just depends on the pore size. So thank you very much. Yes.
What happens if water is contaminated with some chemicals that can damage the filter, or that?
That's a great question. We've never encountered that yet. Maybe we haven't sampled. You're going to sample in the wastewater, right? I don't know what chemicals would do if there was heavy chemicals in the water. We have at times saved the filtrate, so like the liquid that comes out, and people have been interested in screening it for I can't remember what all they were interested in, nutrients though, to see if there was any link. I don't think they actually analyzed the samples, but yeah. Thank you very much. Now you're ready. What did I do with that pointer? Okay, so that's part one. Do we need a break or keep going? I don't need a break. No, I don't need a break.
I know.
And we'll definitely do one here. Go? Okay. And any other questions on how we collect samples before I move on?
Yes.
Before we get back to the lab.
Yeah, I have another question. There's another possibility, but because the idea is very important if you can concentrate on the DNA in the water. Okay, I get this water with the water, and suppose water there are DNA.
Yes.
Okay, perfect. So, and the next step is you go to the filter, right? Now, you get the one with the water and you take that.
Yep.
Okay? So, there is another alternative. Before the filter to concentrate more the DNA, so you can, for instance, you have 1 liter of water. I can concentrate this sample to 500 milliliters, for instance. In this moment, I have more.
So taking a liter and concentrating it to. So I don't see why you couldn't, but I think at the end of the day, you're going to get the same amount of DNA on that filter. So we have at times done pre-processing on samples, but the way we approach it is we try to have fewer steps because there's less opportunity for contamination then, and then the amount of stuff we have to bring and the amount of cleaning that has to be done. So you could do that if you wanted to. We just haven't done it.
Yeah, because the problem is to make it, you have a lot of DNA, I think, from bacteria, for instance. Yeah, maybe the filter is saturated with the bacteria.
Oh, there's definitely bacteria in it, without a doubt. But there's lots of stuff in it, which is why I think this is so cool. And I love going to places and sharing this information, because there's a limit to what we can do, right? There's only so much time, only so much projects you can do. But there's so much potential for these samples to be used by other people for other reasons. And once the samples are collected and filtered and extracted, the cost is quite low in comparison. You know, to go to Panama and collect samples is very expensive. But once the DNA is in the lab, or the filter is in the lab, then the cost goes down.
My last question now. The 0.4 or
0.4. Yep.
Outside of your filter. Why? Is that trade-off between the cost and the result? When you have what is the information to decide why site form you use?
Yep. So you want to use, the goal is to use the smallest possible to capture the most. So the sizes that are manufactured are 0.22. Now why, I don't know, but they manufacture 0.22, 0.45, 0.6, 0.8, 1, 1.2, then it jumps to 3 and 5. So it depends on how turbid the water is. I would love to do 0.22 for everything, but it would take 12 hours to filter a sample if it was really turbid. So here, that was very, very fast. So I would actually recommend a 0.22.
When the water is clear. Okay.
Yep. Yep. Because that took what? 3 minutes?
Yep.
Not 6 hours. But it can take 6 hours if it's really turbid water. Yes.
I have 2 questions.
Okay.
All the DNA remain in the filter, or is there a percentage that are lost?
So any DNA that is, for example, bound to particles that are greater than 0.45 will be on the filter. But if there is like free-floating DNA or smaller particles, so if something is 0.4 or 0.22, it could slip through and be in the filtrate. So that's why you want to use the smallest pore size possible to have the highest likelihood of capturing as much DNA as possible.
And the second, how do the filters behave in the water with oil?
We've never sampled in water with oil from like post oil spill or something. We've never done that. That's a great question.
No, but it's a very contaminated site.
Yeah.
Oil.
Absolutely.
For example, Quintero.
Yeah, because it's a good question, because also, I don't know if it's oil. Yeah, but oil. Okay, but another more typical situation in Chile, in the north, Chile, is the contamination from the copper mine.
Yep. And to my knowledge, I don't know anyone who is and it might exist. The number of eDNA publications has just skyrocketed.
Yeah,
But what is the impact
yes,
in a controlled experiment
yes,
of heavy metals
yes.
or oil on these things?
Yes.
Yeah, very interesting.
What happened with the filter? For instance, if you go to take sample in the north
Mm-hmm.
of Chile, I assure 100% you can experiment this problem.
Mm-hmm.
But this side of the code is very good. Very good. So what happened with the filter?
Yeah, that's a good question.
No?
Because I don't know.
Yes.
That's the stuff I love doing, though, the things that people have no idea. Okay, so after we've come back from our field trips, hopefully by pools, but not always, we're in the lab. And that's where the bulk of the work happens. So the first step, of course, is to extract the DNA. So this is a picture I always love to show of an extremely turbid sample. This is only from 300 milliliters of water. This is from water in Mississippi, where I live. We have extremely turbid, thick water. So it's supposed to be white. All that black is the sediment that was in 300 mils. Okay, so this is the components of the pod. This is a pretty typical filter. This would be 1.5 liters. This is actually from some samples that we collected last year for manatees. And so what we do is, so each sample has its own cutting board that we use, and the entire space is cleaned in between every single sample, and you wear a new pair of gloves in between every single sample. This is cut in half, and the DNA extraction is done on half the filter. The other half of a filter gets archived in the -80, and I am waiting for better technology. So well, there's two reasons we don't just do the whole filter in one go. One, it's students are doing these extractions, and if they accidentally contaminate something, I want to have another half of the filter to go back to. But the other reason is because we may want to process that other half in the future at some point when new technology is available without having to get all new samples, because we have a lot. This shows you a really bad sample. This is absolutely disgusting. There's even some chunks that have gotten onto this particular filter. For me, I prefer seeing something on the filter. Sometimes when the water's very clear, I'm like, I'm very skeptical if there's anything on there, but there is. So we digest it on a heat block. So this is some filters in there. So we use Kaya Shredder columns and the DNeasy blood and tissue kit. So there has been a lot of experimentation on extraction methods. And in my lab, I think we compared 12 or so different extraction variations. And this is what is consistently giving us the best results, this combination. So we heat it, and the filter does not digest, right? We've tried, I was talking about this with the students, we've tried freezing it in liquid nitrogen and crushing it. We've tried scraping it, we've tried bead beating, and then we've tried just digesting it very simply like this. And this gives the best results. So if it's easier, less contamination, that's the way we go. So then we just use the DNeasy kit using fairly standard procedure, except we elute. So the standard with this is eluting your DNA in 200 microliters. We do 50 so that it's more concentrated. These are some gels of extractions, and you can see it's variable. We've got high molecular weight, we've got low molecular weight, but the concentrations are quite good. They can be variable, but we usually get a minimum of 100 nanograms per microliter, upwards of 400. Now, again, remember, this is just DNA from anything in the water. This is phytoplankton, this is bacteria, this is viruses, this is vertebrates, this is just DNA. It doesn't tell us anything about our target DNA at this stage. It just says we got DNA from it.
Sorry, this sample comes from DNA spectrum.
No. Now,
So
when we
a very big
right, we have some big DNA things, and I those are not. This is generally not the DNA we're looking for, but it's DNA I'm sure that somebody is looking for and somebody is interested in. Now, again, when we first started this work, we weren't getting this good of results. This has taken time, tweaking, modifying to get here. And this is with the pods. When we were just rolling the filters and preserving in ethanol, like I feel like we lost a lot of DNA to the ethanol.
Yeah.
And one thing we had talked about was filtering that little bit of ethanol and then trying to recapture the DNA that was just in the ethanol. But we didn't have to do that because then these pods came out. So.
But that is very interesting. The new, that you can get
Such good DNA.
DNA of this DNA from the eDNA from the water.
Yeah, I was shocked. So these were some of the early ones with our pods, and I wanted because the other methods don't give you as nice of DNA for sure. So it's this combination again of the right field methods with pods that are very good at preservation with a optimized extraction method after trying 12 different things, right? At each step, making sure we're doing the best we can, because certainly in the early years, it was more like bleh at the bottom.
No,
it's beautiful.
But this smear is nothing.
Yeah.
Nothing. You have a high weight
yep.
of DNA. So this sample actually is RNA.
No.
No.
No, we do not treat with RNase A.
Why ethanol and not another type of the buffer?
Ah, great question. Ethanol's easy. So we were facing, before these pods came out, we were trying, I'm not sure if you're familiar with Longmire's buffer. That is something that you can ship internationally. So we were looking at that. Ethanol was just because we preserved all of our tissue in ethanol, and everyone in early eDNA work is what they were doing. So we kind of in the beginning, we were like, well, what's everybody else doing? Let's try that. Not really happy with this, and then go back and tweak. But Longmire's buffer also works really well. Well, not as good as this. So I was over the moon when I saw this. Okay. But the bulk of it, right? The bulk of the work, the PCRs. Okay, so I'm going to first talk. So if you pick up any eDNA literature, you're going to see a lot of variation in methods. So I am talking about some specific things today, but if you go and do a search and pick out a random paper, they might use completely different methods. It's very unstandardized at the moment. You can actually use conventional PCR, you can use qPCR, or you can use what we use, which is droplet digital PCR. So I just want to quickly go through the differences in these and how you analyze data, because the droplet digital PCR is very different. Another point is all eDNA PCRs have to be in designated hoods. So this is our hood for our assay development. So this is where we're doing our PCRs with, say, that synthetic DNA or maybe gDNA. And then this is the hood in the ddPCR lab where we actually set up the eDNA PCRs. All right, so let's say you're just using old-fashioned conventional PCR. There's a surprising lot of studies that still use this. So you've got your PCR mixture, right? You stick it in the machine, and then you take your product and maybe you run a gel. Now, I hope that you sequence those products to confirm your species ID. A lot, and this is where I say there's a lot of bad eDNA science. A lot of eDNA studies stop here. So if they have a product, maybe in the assay development, they will sequence one of these to confirm that it is what they think it is. But a lot of them will just stop here. They're like, I have an assay that I got from a paper. I'm amplifying it. Here's my product. But you should sequence it to confirm.
So,
We do not do this. Big X through this.
okay.
Absolutely not.
But it's a specific primer for
Yes.
one species.
Yeah, so here you would design, I'm going to talk about the assay design, but here you would design maybe an assay that's species specific. And then if you had a band, you would just say, oh, look, I got my species. I don't trust that.
Okay, how do you know when it's on a slot, for instance, when you don't have nothing, any band, how do you know it's a negative DNA person, or what the problem with your PCR?
That is a great question.
So
And that's why I don't use this. I also, you know, you'd have to have a lot of product for it to show up here, right? If you have just a tiny amount of product, it's not even going to show up. So I don't trust negative data from conventional PCR personally. My suggestion, if it's okay, very short on budget, and if you had to do this, I would say sequence all of this. Sequence your negatives to see.
But maybe it's not more easy to make a positive control of PCR.
Yes. So, okay. So if you're using conventional PCR, yes, you can have a positive control, and that's where you would use that flagged oligio, where then when you sequence, and that's why you have to sequence, right? Because then if you sequence it and you see, oh, this sample has my inverted tag on it.
Okay.
Yes, yes. But not everybody does that. Some people just say there's a band that's positive, no band that's negative. This is why I say there's a lot of bad eDNA science. Let's say you want to then use, so one step up, conventional is kind of like, if you wanted to do a step up, you can do qPCR. And I know you guys do qPCRs because I've seen your plots last week and this week. So with qPCR, again, you make your mixture, you put it in the machine, but here, right, you have fluorescence. For eDNA, you have, I'm going to talk about assay design, but you could use an internal fluorescently labeled probe, or you could use like a cyber mix if you wanted to as well. And here you've got, right, maybe you've got amplification. If you have your species presence, you're using your species specific assay. Again, though, a lot of folks don't sequence this. If they get a curve, they say it's positive, and if there's no curve, they say it's negative. Some will go the extra step and sequence it, which is what I would always recommend. ddPCR is very different from these other two. So you start with, there's many more steps as well. It takes about 8 hours to do this. So here's your sample. And you first put it into what's called an automated droplet generator. So this is a robot. I saw that you have one in your lab, not this exact one, but you also use the robots. So you set up your PCR, and your plate goes here. And then it picks up the sample from your plate, and it puts it back here in these cassettes. And then it adds this proprietary, expensive, don't know what's in it, oil to each sample. And then it takes the sample and puts it into a new plate. And it generates now a sample that is partitioned into 20,000 nanodroplets. You then take that and you put that into your PCR machine. In this case, for ddPCR, you also have the option of an internally labeled probe or Eva Green. I have used both. I do not like Eva Green. I think it's unreliable. So we always use an internally fluorescent probe. And if you have, if you have amplification in a droplet, that droplet fluoresces. But how do you actually read it? It's just a conventional PCR machine you're using. There's another step. You take the PCR product from the PCR machine and you put it into this droplet reader. Now, this is Bio-Rad. Bio-Rad invented this technology. There are other brands now, QIAGEN and some other ones that exist. They don't call it droplet digital PCR, they just call it digital PCR. I had a rep in my office from one of them trying to convince me, with this, I can process a 90-well plate. With the competitors is quicker, okay? It is faster. However, everyone I've talked to who have used the competitors' equipment has had very inconsistent results. And you can only run 16 samples. So when I told the rep, yes, but how long does it take me then to do 96 samples and it's the exact same time frame, I'm going to stick with what works. Plus, it's very expensive. It's not something you just replace on a whim. I'm going to show when we talk about analysis after the break what the actual software looks like. But this is the data you get. Okay. And just the basics of it. If one of your droplet partitions does not have your target DNA present, it does not fluoresce. And those droplets form this gray band here. So in this particular one, there are 16,388 nanodroplets that do not have target DNA in it. If there is DNA in it, oops, then it fluoresces and it shows up as a blue dot. So here we have 190 detections, 16,388 negatives. So this one had slightly less than 20,000 droplets. Now, how do you know what you're going to use? Conventional PCR, low sensitivity. We did talk, or we kind of hinted at this a little bit with chemicals, but there's sometimes inhibitors in the water. We mostly deal with tannins from plant material, and tannins will basically precipitate out your polymerase and prevent the PCR from occurring. PCR inhibitors are present in water samples, and it's very, very likely that some of those negatives may be false negatives if you use conventional and quantitative PCR. PCR inhibition with droplet digital PCR is minimal because you're partitioning the sample. And I am going to show a video that kind of like shows this in the analysis. Quantification, you have none with conventional PCR. You have relative quantification, but not very good quantification with qPCR. You know, standard curves are problematic. But droplet digital PCR has very precise and accurate quantification. All of this, ultra high sensitivity, no inhibitors, and very good precise and accurate quantification comes at a cost. It's the most expensive of the three options. However, I mean, and that dictates what you're going to use. We all know that. We're all budget limited. Conventional PCR, you could do it in a way that I think is okay, but I don't think it's suitable for rare targets. at all. I think it's suitable for very common things, which do you really need eDNA in the first place then? qPCR is okay if you have a less common species and you only need relative quantification, but you have to be really careful when you're interpreting your results because it's not as sensitive as droplet digital PCR. This is best for rare species and absolute quantification. Because of this, this is what we use in my lab. I'm extremely fortunate in that the University of Southern Mississippi has a grant from the state that is able to purchase and maintain this equipment for our use. So I did not have to buy this machine. I do not have to maintain this machine. And they even cover some of the consumables because that proprietary oil, and as you can imagine, all the pipette tips are proprietary. All the plates are proprietary. So they cover that. We have to pay for the reaction mixtures and some of it, but that puts us in a very good position to use this technology and then make this technology available to collaborators as well. Okay, any questions on that before I talk about assay design? This is everything. If you're doing targeted species, you can have every other component right. And if you don't do this right, your data is not good. Okay, so what do I mean when I say this? So you need species-specific primers. You need primers that match your, I call it target DNA. It doesn't matter what we're talking about. It doesn't matter if we're talking about shark DNA. It doesn't matter if we're talking about a gene in humans, but it's specific to your target. And then we have non-target DNA. Later on, I call it exclusion species. And so in your primers, you have to have base pair differences between your primers and the things you don't want to amplify, right? Simple enough. Maybe throw in 3-4 base pair differences there. All right, this is the steps. This part is so difficult. This is the most challenging part of eDNA work. For some taxa, this can take a year or 2 years to do this part. First step is, to identify a candidate. I'm going to break this down with an example to make it more palatable. You have to figure out what locus are you using. We use mitochondrial DNA for this type of work. So what locus are we going to use? We have to create a genetic reference library with your target and non-target, design your primers and probe, confirm it works, optimize it, then cross-test it in exclusion species, go back to the drawing table if needed, and then validate it. So this is a very long, difficult process. So I'm going to use the example of the large-toothed sawfish because we talked about this yesterday. So you might remember, I talked about this decline that the species has experienced, and our genetic data has showed that there's 2 very distinct lineages, right? And the lineage over here and the diversity in this part of the world is secure. But the diversity and the populations here, which are they are genetically distinct, are at high risk of going extinct. So this is a high priority. Find these species in this area. If they exist, we need to find them, protect them, study them. So this is how all of these things, it may seem disconnected. I do conservation genetics, I do eDNA, but they inform each other. So as a result of this work, we actually launched eDNA studies for this species. And I'll be talking a little bit about those data, the actual results of that work tomorrow. So when we're talking about this species, we're first step, what locus are we going to use? You have to have an understanding of levels of polymorphism in different taxa. So every time we take on a new taxa, it's something new to learn. Okay, what markers should I be using here is the first thing we have to figure out. You want a locus that is conserved within species across their range, because ideally you want it to work across the species range, not just this one location. So in this case, what this means is we need to have a locus that's going to work with DNA from animals in the Indo-West Pacific and with animals from the Atlantic and the East Pacific. So it has to be conserved across these populations. And that's why you have to know about population structure. But you want that locus to be variable in between species. You want it to differ between species because you don't want to accidentally target things you're not interested in. So we call those exclusion species. The other purpose is to, if you sequence the gene, you want to actually know it's your species. So you have to be, it has to be able to differentiate this species from another. So the exclusion species in this case, these are going to be your closely related non-target species. For large-toothed sawfish, this means our exclusion species are 4 other species of sawfish, guitarfish, giant wedgefish, and wedgefishes. These are all those rhino rays. So these are across different, within genre, within family, within order. So we usually test up to that taxonomic level. So the key is you want a locus that is not too polymorphic, but you also want it to be not so conserved that it also amplifies other species. It has to be just right. In sharks and rays, what's primarily been used is 12S, 16S, and ND2. These are all coding. And this is what we've been using in other taxa. Manatees and freshwater fish and freshwater turtles, we've used cytochrome B. For sea turtles and amphibians, we're using ND2, and for marine bony fish, we're using ND5. So like I just took on the amphibians and I went, okay, I don't know anything about amphibian genetics, and that's the first thing you research and try to learn about. For large-toothed sawfish, we use 12S. There's conveniently a lot of 12S data available, which I actually generated 18 years ago during my graduate work. Then we create a reference library of that gene. So we've got our target species on here. This is a full mitogenome from Australia. And then we have a tissue sample from an animal from Ecuador. And this is just an example. This is obviously a much larger data set than this, but this just highlights that we have samples from across the range. And this is another one. And then we have our non-target, so other species of sawfish, wedgefish, giant guitarfish, and guitarfish. So this is the 12S gene for these things. And there's a little bit of variability. It's not a crazy amount of variability for how different these species are. But remember, this is why you have to understand mutation rates. These things have very slow mutation rates. So we might not see as much variation as you would see in other critters. So when we actually go into design primers and probes, you use the general rules for primers and probe design, same as what you guys use. But you have to target a small fragment because, yes.
Perform the 18A sequences of the water sample directing and not go to know and become sequencing.
Can you repeat the question? I'm not sure I understand. I don't know if I understand.
No. Utilizar una secuenciación del 18S de la muestra total y no de la secuenciación del amplicón del PCR. ¿Cómo va a ser la secuenciación de la muestra total? Lo filtra y el filtro, la extracción de DNA la pasa directamente a la secuenciación en vez de amplificar el gen que anda buscando directamente.
Pero el DNA lo pasa a una resolución.
¿Lo van a secuenciar?
Claro, pero quieres hacer pero quieres una resolución del genoma.
Es lo mismo que un metagenómico.
Pero necesitas
El backbio no tampoco necesitan.
pero necesitas amplificar. Necesitas amplificar para secuenciar. No quieres construir una librería. Necesitas amplificar para secuenciar. Just maybe it's a confusion.
Okay.
From
Yes, so we have to develop everything. There's so with this step, we've got the filter, we've got the DNA extract, but this part, we have to develop the methods to actually screen for our target. I know in a lot of places, primers and things already exist, and you just order these things that already exist. We have to do everything from scratch. Yes.
What happens with species that are not you also do?
So do you mean like if a sequence isn't available on GenBank? Is that what you mean? Like we don't have sequences?
But for example, I don't know, viruses that we don't know
Okay. Yeah, I think that's the question. So when we do this reference library, and this is why it takes so long, maybe things aren't available. We generate it ourselves. So if for example, if there isn't anything available for some of these, I call somebody up and I say, can you get me some tissue or DNA from this? And they send it. And then we have to, then we just usually use like a universal primer and we sequence it and we generate this ourselves. So for a lot of the species we study, just getting this reference, one slide is 6 months of work potentially.
It's just one example.
Yeah.
There are a lot of more species.
Yep. Yes. This is just to make it more digestible. Yes. So this here, this is an example of a guitar fish. Here we have a wedge fish, giant guitar fish, sawfish. So I just threw some things in there as an example. These tables are huge. And yes, if it's not available on GenBank, we have to generate it. We can't just say, this isn't available, so we're not including it. That's not an option.
So that methodology also can be for differentiate not just different mutations. So
Yes. Yep. So here we can sort of see, there's some conserved areas and then there's some more variable areas, but really not very many, honestly. 12S is very conserved within species in this taxonomic group, and it's variable between species, but it's not one of the most variable regions between species, certainly. But if you went to a more polymorphic marker, then you might have too much variability across the range of your target species. And I want something that a collaborator I want something I can use in the East Pacific, the West Atlantic, the East Atlantic, and the Indo-West Pacific. So it's about having thinking about larger scope of how this can be used and where and by whom, because it's not just for me. It could be for other researchers as well. All right, so for the primer, how we actually design it. So maybe now we've selected our locus because we're like, okay, there's some variability here, that's enough. The fragment has to be small, between 100 and 250 base pairs. There's 2 reasons for this. Remember that DNA is degrading. And if you go for a really big fragment, the likelihood that fragment is intact is lower. So technically, the smaller you go, you should increase your detection probabilities. And I have a student who's working in, I'm not a stats person like that. So he's working on detection probabilities and all of these variables. The other thing is ddPCR is really for small fragments. It's not recommended for things, for example, larger than 300 base pairs. They recommend 250 base pair amplicons and then your primers. And then I said, I don't like Eva Green. We use internal probes. And the probes have to have a melting temperature that's 6 to 8 degrees C higher than the primers. And you have to design those primers and probes in those conserved areas within species and maximizing base pair differences between species. When you put all of this together, this is really challenging. It is a Tetris to do this. And then, of course, you have all those things where you order primers and on paper they should be fine, and then you try them and then they're garbage. That happens too, which is fun. So here's our example of, again, it's just a shortened, truncated example. So this is the forward primer for this species. This is the primer. This is the target. This is target. And all of these are non-target. So we've got variation in our primer and the species that we don't want to amplify DNA from.
That is 12, please.
Mm-hmm. Yep.
I don't know if you have considered include in the factor of the secondary structure because you have stem and loop. Maybe it's not that you show now. You have many gaps. So this is
yeah, indel. These are indels.
Or indel.
Yeah.
Indel. So maybe you have loop sector. The problem with this gene from this ribosomal gene from the micromolecule
yep.
from nuclear, problem is this gene there are secondary structure. You have loop, just single train and stem with a double chain. So in this loop, the possibility of mutation is very high because it's just one chain, nothing more. But in a stem, but you have two chains, the capacity of mutation is very low.
Right.
So for this reason, in this typical gene alignment for this gene, when comparing several species, you have sector very conservative. There are stem.
Yep.
And sector with a high variability. There are loop. The problem with put the primer in the loop is this sector have high mutation rate. So I don't know if you have considered this aspect.
Yes. So 12S is really well described for this taxonomic group. Yeah. Maybe not others, but we have a lot of 12S data to be able to, and in mitogenomes, where we can see that there's more variable areas and then there's more conserved areas. So that's why we're very particular about where we put these primers. So within this gene, we may have had 5 different sets. This is the set that works.
Yeah, I understand.
Yeah, but the polymorphism is correct. Is the polymorphism we see here within species is okay. Like we can have some polymorphism, not in the primers and probe, but within the fragment, we can have some polymorphism within species.
I understand, but my own suggestion is, okay, cool. You have all the alignment of this gene or whatever. So my my suggestion is just to delimitate sector. For instance, from this column to another column is one stem. From this column from another column is one loop. Just to confirm that the conservation sector is why is come from one stem sector. But just,
Yep.
for instance, you have here a very conserved sector. Yeah.
Yeah.
You know that. By why?
You mean within species or between?
Yeah, no.
Well,
Why?
it's probably a conserved part of the gene because it's coding.
Why? No, why?
I don't know.
If you are working with this gene, maybe this is a stem.
It might be.
Yeah.
Yeah.
So the idea when you make that is say that is a loop.
Yeah.
That is a stem.
Yeah. But it depends on the taxa.
No, what
because the mutation rate's different.
But it's very conservative.
Yes.
So the secondary structure of this gene is very conservative. So I 100%.
You think that's the stem?
That is a stem.
I'll check.
Yeah.
Because within the sharks and rays for 12S, most of 12S within species is very conserved.
Yes.
The whole thing within most sharks and rays, because we've got 3 sequences from remember I showed that graph to show how genetically distinct they were between different oceans. So we've got Indo-West Pacific and Atlantic Pacific are very different genetically on other genes. At 12S, they're conserved across most of the gene.
Yeah,
there's a little bit of variation, but not a lot. It's like you could never use 12S to differentiate populations because it's too conserved.
The shark have the group of sharks have a mitochondrial genome with a very, very low potential.
Correct.
In comparison with another.
Correct. The lowest of any vertebrate, which can actually make this very challenging because something that's super variable in 12S, freshwater fish are quite variable, is not variable here with this taxa. So that's why in this stage, where is it? This stage, that's why we're targeting different things across different taxa. And the first thing we have to learn about is the mutation rate of different genes. For me, sharks and rays are easy because it's what I know the best, because I've worked with many different genes in this taxa. But when I go to something I'm less familiar with, then we have to learn about, and we usually actually create reference libraries for multiple genes and then see, because you'll notice 12S is the only one we use for sharks and rays. And it's because it's within species, across oceans, very conserved.
Yes.
So, yeah. So when you add all these things together, it's really hard. And this is what I wish it'd be interesting. Like, so we do this manually. We talked about this yesterday. We do this manually. There is no programs for this. This whole component of variability of loci plus, needing to amplify in some things and not others. It's very confusing. So here's our forward and reverse in this case. And here is our internal probe. You can see the amplicon is very short, right? Very short. You have an assay, you have to test it in your target species. So this shows a test for our large tooth sawfish. And each of these dots is a positive. This is an eDNA sample. So that's why it's very low. There's only 1, 2, 3, 4, 5, 6, 7. 7 of those droplets of 18,000 droplets had DNA. Now this isn't sawfish, but I just want to put this up there so you can see that this is possible, because this is something new. You can target multiple targets in a single well. So this is our Seriola assay. This is that negative, it's the same exact concept. This is our negative band. So all of these droplets have nothing in them. And we have, in this plot here, we only have 6 FAM as our label. Here now we have 2 types of fluorescence, and one is red here. This is FAM, and this is Vic, which is red. So now we have a different plot. So this is our negative, and we've got on the Vic fluorescence, we've got 1 species here of Seriola, a second species here, and then on the FAM fluorescence, we have a 3rd species here and a 4th species here. Because for this particular work, I'll talk about this tomorrow, we have interest in differentiating between species in water samples. So this is the first use of 4 different species. Technically, you're supposed to be able to get up with upwards of 8. That would be exceptionally challenging. The way this was designed was a little bit different, so I just want to mention it because this is bony fish. This species has a lot more variability than our sharks and rays. And so for this one, what we actually did was, instead of designing species-specific primers and a species-specific probe, for sharks and rays, we have to do that because sometimes we can only get 1 or 2 base pair differences in a primer. That's not enough. That's not enough. So we have to have as many as we can get in each primer and the probe. For this species and this locus, we have 1 set of primers that amplify all 4 species. So they're universal for 4 Seriola species, but they don't amplify any other Seriola species. Then we have a probe that is species specific. And so we have 4 species specific probes because this was variable enough to make that work. There are, I think, 7 or 8 base pair differences in these probes. I'll be talking about this study tomorrow, but I just wanted to show you this because this ddPCR, one thing I didn't mention is when we first started using this for eDNA, no one had ever used this technology for eDNA. Do you know what it's used for? Any guesses? Gene expression. So apparently, so the lab that we were using a machine in worked in humans. Something with I don't know the details of their work, but it was with childhood obesity and genetic factors. And they looked at gene expression, rare target gene expression. So when we decided to try to use this for eDNA, that was kind of crazy and strange. So this is not the typical use of this technology. Most of the time, so people will tell me, I don't have access to this machine. I don't have access to this machine. And I always tell them, do you have a local research medical hospital or a research school that does medicine? And many of those schools have these machines. They're just not typically used as routine yet because they're too expensive in ecology or molecular biology. Okay, then, so once you know it's working in your species, so you've confirmed it works, then you have to make sure it doesn't work in anything else. Because if it amplifies something else, it's not useful to you. And so this is an example of a plot with exclusion species, species we expect not to see amplification. And we don't see any blue dots on here. Then you want to validate it. A lot of people skip this step. So you want to validate it with a known eDNA positive if possible. There's ways to simulate this. If not, I did a simulated way for manatee work. So for sawfish work, we were lucky enough to be able to go out and we collected a baby sawfish and we put it in a tank with a ton of permits. And then we collected water samples at intervals. Again, at this time, no one had done this before. We didn't know what to expect. This is just our negative control. For the PCR, the other negative controls just aren't included in this graphic. We collected water from known habitat. So we went to habitat. We knew there were sawfish there because we had those radio trackers that you put in the water and they can hear tagged sawfish. We did that. We knew they were there. We took a water sample, but we had no idea how close we had to be to the animal. We didn't know if we would be able to detect it from presence. So that's why we took one and put it in a tank. This was actually the signature from the habitat where we knew there were sawfish. So there were all of these positive droplets. I think it was something like 700 maybe 700 nanograms per microliter of target DNA from water. Our tank experiment just blew up the machine. This is too much. This is too much target for ddPCR. If I wanted more accurate quantification, I would do a dilution factor on this, because here we see after it says 0 minutes, but it kind of thrashed around. So it might've been 1 minute. It had shed, and this water here was the tank water. So we took the ambient water, we tested it, we put it in the tank, and then we added the sawfish to it. So this difference is what was shed by that animal in roughly a minute. And then after 30 minutes, almost every single droplet, it was just 3,000 nanograms per microliter. Crazy, crazy high. Okay. So at this point, we can have a quick break.
Yes, maybe.
Yep. I'm going to run to the bathroom. I'm just going to go to the bathroom.
No.
All right, I would say that that was like this part was probably the heaviest, maybe the hardest. So even though data analysis is like, oh, this is going to be the challenging part, the data analysis isn't too bad for the stuff we've done to date. So we'll talk about how we actually analyze data. I'll show you what the software looks like and how we interpret it, and then how this overall approach has impacted conservation. So I just put this up one more time as a reiteration. Here's our ddPCR product, because we're going to be looking at a lot of these plots. If the droplet does not have the target DNA present, the dot is gray and forms this dense band of negatives. And then if it has the product, it's going to fluoresce because it has that fluorescent label, and it is red. In this particular one, it's 6-FAM as a blue dot. And this is one of our actual samples. This is from a validation, but it doesn't always look like this, as you're going to see. I'm going to show you all the variation in the plots that we get. So the analysis software that we use, this comes with Bio-Rad, with the system. It's called QuantaSoft. And this is the first screen that you get. And this is your plate. So it's quite similar to qPCR, right? You've got each sample, you label it, you write what locus it is, you write maybe if it's a negative, or if it's a sample, or if it's a blank well. And we try to avoid that. We try to have all of our samples on there, and the analysis. So you enter all of this into your plate setup right here, and then you click run, and this is in that droplet reader machine. And so what it does, and this makes it really different from qPCR, is it'll pull the sample and then it counts the droplets. This entire process for a plate takes 3.5 hours. Counts the droplets, but this is the key. It discards the sample. So you don't have anything left over. After your sample is read, you just have an empty plate. So it actually destroys the sample that you have, which is a con because you can't sequence it. That's why assay development is everything. You have to be overly rigorous with your assay development with ddPCR compared to qPCR, where you can just sequence it. Okay, the analysis. So the computer does an analysis, and I have a video that I'll show you in a minute, which I think it explains it fairly well because it's a bit complex. But it really just shows you how it's different from qPCR when you're looking for rare targets or rare mutations. And so the analysis, it does what's called rare event detection. So it looks for rare targets or rare mutations. And it also has, so this will give you positive or negative. And it also has absolute quantification using Poisson statistics, and that is going to be shown in the video. Can you play the video?
Now, they are